Thank you so much, everyone, for joining us this evening or late this afternoon, depending on where you are. My name is Dorothea Lance. I'm the Director of Community Engagement with PWSA USA. This is a joint webinar between PWSA and FPWR, and we are really excited to be joined today or this evening or late afternoon by Aardvark Therapeutics. We have Dr. Tien Lee, who is the Chief Executive Officer of Aardvark, and Mansi Jamin, who is the Chief Medical Officer of Aardvark, who are going to be sharing some wonderful details about the HERO study to treat hyperphagia and PWS and some exciting new developments in their study. And with that, I will go ahead, put myself on mute, and turn it over to our stars of this evening's show.
I really appreciate it, Dorothea. Thank you. Really appreciate the opportunity. So I'll go back and forth between Mansi and I, but just a quick introduction. I'm Tien. I'm the founder CEO of Aardvark, medical doctor by training, and we started Aardvark in 2017, and very happy that we're at this point here. And maybe Mansi, a quick intro.
Sure. Hi, everyone. Thank you for being here and having us. I'm Mansi Jamin. I am the Chief Medical Officer here at Aardvark. I'm also a pediatric endocrinologist, and it's very meaningful to be part of this company and being able to do this work because it serves the patients that I used to see. So thanks for having us here.
Great. So since we're sharing the presentation of the slides, I'll just do the first few. And Aardvark's a publicly traded company, so this is a random, this is the usual disclosures for that. As I mentioned, we started the company in 2017, and the lead product is a oral small molecule drug that's orally delivered, and we believe it reduces hunger as one of its primary mechanisms. And I'll go more into that in the next few slides. But we actually elucidated this very interesting drug that signals gut-brain signaling, and we actually over time, when we understood what our drug was doing, it seemed very evident that Prader-Willi syndrome would be a matching therapeutic target for what this drug was doing. So we have a lot of optimism in terms of what it could do therapeutically. And presently, we're in the midst of a global phase 3, potentially registrational trial called the HERO trial, which stands for Hunger Elimination or Reduction Objective. Next slide, please, Mansi. The drug actually has some unique properties. It's actually a gut-restricted drug, so the majority of the drug actually stays within the gut and is not systemically absorbed to a high degree. About 1% gets systemically absorbed. And normally, this is a feature of a drug that is not usual, but in our case, we're targeting cells in the gut to release gut peptide hormones that can regulate and convey satiety for hunger. So it's hitting bitter taste receptors actually in your gut. You do have taste receptors, not just in the mouth, but in your gut. The drug is coated so you don't taste the drug, and when you swallow it, it releases its contents in the gut. And by virtue of this mechanism, it's signaling. I mean, I think you may have heard about the vagus nerve. There's actually two-way communication. There's brain to the gut, but there's also gut to the brain traffic. And what we're doing with our drug is to increase that signal from the gut to the brain to convey satiety from hunger. Is there another bullet? Yes. So we have a really good confidence going into our phase 3 because we did a phase 2 study with Prader-Willi individuals age 17 and up. And it was very well tolerated. We've over 70 patients before we even got into phase 3, and the safety assurance was very high for us, given what we've seen or didn't see. And we had a lot of encouraging signals of effect in our phase 2 study, which gave us confidence to go into the phase 3. And when we started the phase 3, I think Mansi will go into this further, but we had started with ages 13 and up as the inclusion criteria, but we just got alignment with the FDA to now incrementally expand the ages to 10 years old and up, and we have intention to try to further include even younger patients still. And again, Mansi will go into that in the later slides. Next slide, please. So really, the centerpiece of our story is the dichotomy between hunger and appetite. A lot of folks might not conceptually think of these as separated, maybe different extremes along the same continuum. But the way we're looking at the drivers of eating behavior is that appetite is a reward-based drive. Your brain is trying to go after something it wants. So, for instance, if you like ice cream, I like ice cream. That's always thinking of this as the analogy, but your brain is telling you to eat more ice cream because it desires the reward of that ice cream. Common obesity drugs now that the GLP-1 drugs, they can diminish the appeal of food, but they don't as much directly address the hunger element. And what hunger is the opposite end of the spectrum, but where you think of appetites like the carrot and hunger is the stick. It's the feeling you get when you skip a meal or two or three and you have this fasting and it is very, very uncomfortable. And when your drive to eat is driven by hunger, food quality becomes less important and you want to eat anything to kind of push down that uncomfortable feeling of hunger. And we believe this is where ARD-101 is particularly useful because we're activating this gut-brain signal, which we believe turns off hunger, which is the one of the driver of hyperphagia in Prader-Willi. We think that there are lots of points of evidence that show that it's a hunger-driven drive, not a appetite-driven one, which is why we are very optimistic about deploying ARD-101 in this context. Next slide, please. And another reason why we selected Prader-Willi syndrome as a therapeutic target for our drug was because gut-brain signaling has a lot to do with more than just relieving the feeling of hunger. This gut-brain signal also has anti-anxiety effects, can improve body composition, can normalize and regulate gut transit. As you know, in Prader-Willi syndrome, many individuals have delayed gut transit and constipation. And we think that the gut-brain signal, including that driven by this gut peptide hormone called CCK, can help normalize that and also can help reduce inflammation. And this is another manifestation in Prader-Willi syndrome individuals where a lot of individuals have a lot of inflammation in their clinical picture. And we think that activating this gut-brain signal can potentially address multiple avenues of challenge for those with Prader-Willi syndrome. Next slide, please. Oh, and then this is, I'll just turn this over to Monsi.
Thanks, Stan. So now we'll go into details with our phase 2 study and what led us to our phase 3 and what we're anticipating as well. So Tian talked to you about the background, kind of what we expect from a scientific perspective, but this is what we saw in our phase 2 trial. So ARD-101 was tested in patients who were 17 years and older for 28 days. We looked at hunger and hunger changes using the hyperphagia questionnaire, the HQ-CT9. And you can see on the right side that we actually had a change on average over 8 points on this questionnaire, which is an important finding for us. And we also had improved early signs of fat and muscle balance. And this was, again, really important because within PWS, as you all know, there is lower muscle tone. So what we wanted to show is that we can actually preserve that. And these were great early signals to see. So also from a safety standpoint, which is also essential, is that we have dosed over approximately 70 people with ARD-101, and we've seen a fairly clean safety profile. What does that mean? So majority of the side effects were mild and all resolved. There are no known problems with other PWS medications while on ARD-101. And also there aren't any real risks to liver, kidney, or the immune system. All important as you kind of look at the bigger picture of what we're trying to do. Does it work? Is it safe? Important questions to think about. So that brings us to where we are now, the HERO phase 3 trial design, which was informed by our phase 2. So if you can look here, this is our schematic. So patients who have PWS with hyperphagia, it's a primary requirement to participate in the study. Previously, we were 13 years and older, and Tian, as mentioned, which is what we're super excited about, is now we're able to go to 10 and older. And that's new guidance that we've gotten, and that's what we're incorporating now in the trial sites in the US. And as you can see, there are 2 treatment arms, which means that people get active drug, which is ARD-101, or placebo. And the total number that we're trying to achieve in this study is approximately 90. And I'll show you in the next slide that we also have an open label extension. So the duration of this phase 3 study is 12 weeks, where you're given either active or placebo, but then everyone is allowed to come into the open label trial, which is now approximately a year, and everyone gets active ARD-101 study drug. So it's a short period of time. We're in the phase 3 parent study, as we call it, but then everyone gets the option to really get this active drug for approximately a year. And we're trying to actually give that as long as it's needed until we have approval, but we're starting out with 1 year. And open label extension studies are often done in a clinic after a main clinical trial to really ensure safety follow-up as well, and having a longer term availability of the drug to patients. And again, we're looking at the HQCT9 as our primary endpoint that we have also done for our phase 2 trial. So phase 3 as well as open label extension. Questions that we often get, what are the other requirements to be in this clinical trial? So outside of having PWS and hyperphagia, and of course, I mentioned that now you could be 10 years and older, specifically in the United States. We require that the same caregiver is part of all the visits, just because these are questionnaires. So we want to have the same oversight of the patient involved by the same caregiver at these visits. And I'll kind of go into how we actually designed this trial to decrease the burden of clinical trials. And then main requirements are complete questionnaires, attend these visits. And then one thing to also note is that we have required 30-day washout for any investigational drug before screening. So if you were involved in any of the clinical trials or had an investigational drug or device on board, then we require 30 days from the last time you took that in order to participate in our trial. So it's only 30 days, but then you're able to participate. So we really wanted to make this protocol with minimal burden, recognizing that the burden of PWS and adding a trial on top of that. So we simplified the schedule. So there's only 4 clinic visits and 3 telehealth visits. There's also no fasting. Again, trying to decrease the burden of participating in the trial, and we actually remove fasting from this. It's also, as I mentioned, important to have the same caregiver, but that also actually helps with keeping the same routine during the trial as well. But it also helps with some of the endpoints that we're looking at. We are covering travel and lodging as well. And that was really important to us because we recognize that it is a participation in a trial that can be strenuous, and you may have to travel different distances. And so we wanted to make sure that burden was decreased. Again, travel and lodging is covered. After the phase 3 has been completed, you can join the OLE, and those are the next steps. And we will be providing support during that trial as well. Again, where are we located for these clinical trials? So you can't really tell exactly these spots on this map, but I'm going to provide you on the next slide a QR code that you can use to go to our main websites that outline where we are. Also, clinicaltrials.gov. Some of you may be familiar with that. We'll have exact sites and their contact information on there as well. And this is for global. So as we mentioned, we're a global trial. We have several sites in the United States as well. And I will show you on this next slide, and I want to stay on here just for a few seconds, and I'll come back to this at the end of the presentation, so you can go back if you didn't get a chance to look. But this is the QR codes that will take you directly to our websites. So first, it's the hero4pws.com. You can also write that down or type that into your phone or computer. That'll take you to our primary website for this study. That'll help guide you through where are we located and how to get connected to a trial site. We also, on the right side, will take you to the clinicaltrials.gov listing as well, both if you look at the phase 3 study as well as the open label extension. So all the sites that are participating in the phase 3 will also be part of the open label extension. So that is just outlining kind of what are the requirements. I'll take a few more seconds and then I'll move forward with the next slide. Okay. We'll move on. So our website was made to be easy to navigate. There's 2 languages available at this time. It kind of redirects you to fill out a very short form to see if your family member may qualify for the study. It will also connect you to LeapCure, which I'll discuss later. You may or may not be familiar of us utilizing a partnership that we have with LeapCure, but I'll talk you through what they are on the next slide. Again, it's a fairly straightforward site. We didn't want to make it too complex. We want you to be able to understand kind of what are the eligibility requirements, the study details, where we're located, easy to access. And we kept that in mind just because we know sites can get super complicated with too much information. So what is LeapCure? LeapCure essentially is a concierge service we are partnering with. They provide the one-on-one support for the HERO study. So again, we really wanted to make this a seamless process for everyone that wanted to be involved in the study. So from the moment that you're interested to the moment that you are activated at a site and enrolled in a site, this is part of the process. And so either you're directly calling the site because you already have a provider at the site and you're connecting with them that way, or if you don't have a local site available to you but are still really interested in participating, LeapCure is a concierge service that if you go to our website, will be connected after that short form. If you qualify, then they'll connect with you by phone, email, and or whatever way you prefer to be contacted. And then what they do is they identify a site that you can connect with and travel to, and they will be there with you until the moment that you are screened in. And that's just to keep the turnaround time less for you, so you don't have to do as much follow-up and make the process as easy as possible. So if you hear LeapCure, they are working with us, and we're very lucky to have a partnership with them just because they have provided a kind of a seamless a transition from interest to enrollment for us, for patients who really want to participate but don't have local sites. I don't have many more slides, but I think it would be amiss if I didn't actually talk about how honored we are to work alongside the PWS advocacy community. We recognize their compassion, their insight, the collaboration. It's been instrumental in advancing ARD 101, specifically for those living with PWS, but also for so many other clinical trial opportunities. So again, we're so excited to be part of this community and share the work we do for the patients that we care so deeply about. And lastly, but not least, we're grateful to the participants and the families. We see the effort it takes to join and stay in these studies, your appointments, travel, the routines that you have to break and change. And we're so thankful for the sacrifices that you've made to help advance the work we're doing for patients with PWS. So that's it for us right now. Happy to answer questions. And thank you again so much for allowing us to be here to talk to you today. And I'm just going to go back to this slide just in case you want to get that QR code again.
Nancy, thank you so much. And Tian, thank you as well. I know that Susan Hedstrom from FPWR is also here. If you wanted to take a moment to say hi to everyone, Susan, please feel free to hop in. We have just a small handful of questions, but I'll give Susan a chance to say hi to everybody.
Oh, Dorothea, you're wonderful, but I don't need to take a minute away from this presentation. Mansi, Tian, thank you so much. I really appreciated it so far. Let's get right into the Q&A.
Okay, sounds great. Thank you. So our first question is fairly free of symptoms of the 70 people tested. What were the number that experienced symptoms? So I'm thinking this is part of your phase 2 trial question. Do you have an estimated amount of people that experienced any side effects or symptoms?
Sure. So, and I think it's important just the PWS, even though 70 plus means that all patients ever exposed, including some of the other trials that we've done and the early healthy volunteer studies we've done. But in the PWS population specifically, and we've disclosed this publicly, and you may be able to even find some of our previous presentations, but we had two parts to our phase 2 study, part 1 and part 2. Part 1 was 200 milligrams dosing twice daily, so lower dosing. And then part 2 was the dosing that we're actually using in our phase 3 study. And what I can say is to focus on the part 2 only because it's really kind of more apples to apples with the dosing that we have. We had 3 AEs, so adverse events or side effects noted, and so 3 patients, and all 3 resolved, and all 3 actually completed the study and without any deterrence. So one was irritability, one was actually diarrhea, which we thought was a very interesting thing to see, given that there is so much constipation within the PWS population. And you will see in the phase 3, we're actually monitoring for that to see if we're actually improving gut transit time. And we anticipate that that's possible, just given what we know about the mechanism of our drug. But again, very interesting. It was 1 patient, but interesting to see, and definitely something that we're following as we move forward.
Thank you so much. Our next question, is there blood work involved? So I guess that's pretty basic. Is there blood work involved in the clinical trial?
Yes, there is. I wish that we would not have to have any, but it's really important to have those metrics just to understand safety. It's something that's required by almost all regulatory agencies.
Perfect. Can you go over again how the drug is targeted to suppress CCK, but also improves atonia? What were the measurements of success on the last trial, and were weights gathered prior to and after the trial?
Dan, why don't you start with this, and I'll answer the second half.
Okay, so CCK is very so our drug is hitting bitter taste receptors on enteroendocrine cells. These are the cells that release a number of hormones in response to food, but they also can be triggered by these bitter taste receptor agonists, as we call them. And then it's inducing these cells to secrete a panel of these gut peptide hormones locally, and then it triggers the natural signal to turn off hunger. And this is something that you can see in experimental models and so forth, that we by amplifying this signal, you can kind of tamp down the hunger. We also saw a lot of data, like for instance, we had DEXA data in our Prader-Willi trial, and even 28 days we saw body fat percentage decreasing by 1.5%, and lean body mass was actually had a gain of 2% just in that 28-day period. And the reason we were looking for it in the first place was because our preclinical animal data was very suggestive that our drug would be helpful in that arena, so that we weren't looking arbitrarily. We were looking because we expected to see something there. And all the other things that seem, like the different, like the anti-inflammation and the anxiety, we think it's actually very correlated to this exact pathway as well too. So it's all consistent with our expectation, and we actually are very hopeful that we'll see that as well. And I don't want to miss another part of the question. Just if there's I feel like there was a second part to it, I might not have addressed fully.
The first part was, can you go over how the drug was targeted to suppress CCK but also improve atonia, and what were the measurements of success on the last trial, and were weights gathered prior to and after? Right.
So we didn't focus on the weight. We didn't see that much of a change. However, that being said, I think the families that were involved in our trial, they had a pretty strict guidance on food access, but the one we're going for is that feeling of hunger. I think the calorie restriction wasn't much of a thing. If you remember, this drug is not diminishing appetite; it's diminishing the hunger. So the food still has its appeal. So we actually may not, in the context of normal BMI, want to drive too much weight loss, but we do want to affect the hyperphagia. So HQ-CT score was the primary measuring tool for our phase 2. We didn't formally measure anxiety, even though anecdotally we saw lots of encouraging anecdotes about decreased anxiety. We will be formally measuring that in the phase 3 with the PAD-Q. We also will be measuring other gut hormones that are related. And CCK, by the way, we're trying to increase local CCK in the gut level, not decrease it, because we think CCK is one of the drivers that give you satiety. The natural way most people have CCK release is with eating fats, and fats will secrete CCK. That turns off your hunger. That's why if you eat fatty foods, it actually can keep you more satisfied for a longer period of time. Our drug is actually increasing that localized expression at the gut level, and that's what we're trying to do. But a lot of the things that's happening with our drug are localized to the gut. But there are other hints of biomarkers like ghrelin we think that could be useful in tracking how much we're functionally decreasing this hunger perception. And again, if I miss something, I think Mansi will cover. I want to make sure I fully address all the questions.
Yeah. And the last one, I think were weights gathered prior to and after the trial. And so we did, and Tien kind of mentioned this as well. So what we actually had more than half kind of in the normal BMI range, which is what we anticipate just because families and caregivers are mindful of intake and kind of monitoring that. So those that participate in clinical trials, we don't expect to have a majority that have extremely high BMI. And that's true to kind of what's available now in terms of control. So that's why this HQCT9 is an important indicator for us as a measurement of hyperphagia. And like Tien said, we're not trying to make it not taste good or unappetizing. We just want you to not to want to eat as often.
Thank you. That's actually a perfect segue into our next question, which is, can you talk a little bit about using the HQCT as your primary endpoint? And are there any concerns about using the HQCT after the failure of the Acadia study using the same tool?
So I can answer part of this, Tien, if you want to jump in, you can too as well. But I can say this. So the HQCT9, it's not a perfect measurement, right? Because it is subjective. It is a questionnaire, but it's all we have that's been validated by the FDA to use as a primary endpoint. And what does that mean? It's gone through all the rigorous training and quality requirements that is needed for an assessment tool in general. So, and there's a lot that goes into validating any tool that's used in clinical trials, and months of it. And so it's the only version that's available. And this is from like Zafgen days when it was developed and then validated as a tool. Several companies have used it, obviously in the PW space as their primary endpoint. Some have had positive results and some have not. So I don't know if it speaks to the tool itself. It just speaks to, I can just speak to the subjectivity of it and how to control for that. And so, which is one of the things that we've tried to do in our trial. So limit the variation in the caregiver who's filling them out. So there's consistency, because if that same caregiver is with the child, the patient, then they'll be able to see changes over time, which keeps that accuracy and consistency there. If you change the caregiver, obviously you lose kind of that baseline versus where you are in the time point that you're filling that questionnaire out. So for us, it's more about keeping that consistency there, even though it's a subjective score. And I wish there was something else. I wish there was a blood test. I wish there was another quick, I don't know, indicator that shows that you've changed your hyperphagia of some sort, but we don't have that yet. So it's all that's available to use. In terms of Acadia, it is really unfortunate as a physician to not give more options to patients with PWS. It's heartbreaking, and I wish that we could bring more therapies forward, but it is what we have to use. And so you just minimize the variations and variability of the tool that you have to use. And I guess that's the only answer I can give right now. I don't know if Tien, you have anything else to add, but.
Sure. I mean, in a certain way, we're constrained by what is the validated endpoint that the FDA is communicating is important. So HQCT is the primary endpoint, but we actually are very optimistic that we'll have a lot of positive signals in our secondary endpoints too, which include the clinical global impression, which ultimately we're hopeful that will change, and we're very optimistic about that because we think our drug could help alleviate not just the hunger but the anxiety, inflammation, and all those things cumulatively may drive an improved score in that parameter. We unfortunately didn't formally measure that in the phase 2, but we are very optimistic that there's rationale to see a very suggestive signal in a phase 3 if our thesis is correct, which we believe we are. And also want to remind everyone that our phase 2 was limited to just 28 days of treatment. And the reason why we did that was not because we arbitrarily chose that, because at the time the company was less well capitalized, and we only had the requisite the studies done to enable 28 days of dosing. Now we can dose theoretically in perpetuity because we've conducted all the longer term tox studies with a high assurance. But at the time we could only dose for 28 days. So as encouraging as the signals we've seen and as robust as we think we've seen in terms of the effects of the drug, it's only within the first 28 days we already saw those. So we're very much looking forward to see further improving with longer treatment, which we think directionally there's lots of reasons to be hopeful that it could even be more beneficial than what we've even seen in that 28-day period.
Perfect. Thank you so much. Are you all able to describe the new open label extension phase of the trial a little bit further?
Yeah, so those that complete the phase 3 trial are eligible to participate in the open label extension. Open label extension is approximately 1 year in duration at this time. Again, like I mentioned, as a company and as Tian and myself being physicians, we want this drug to be available to everyone so as long as possible until approval. But right now it's 1 year. There are several spread out visits that we will cover in terms of lodging, travel, to allow for you all to be able to make it. But it is, and in terms of the visit schedule, that's will be shared at a later time. But most of this is available on the clinical trials website in terms of just the duration of the trial itself. So you finish the phase 3 study after 12 weeks, and then at the end of that, you can opt in to go into the open label. You go into the open label, and it has a few labs that you have to take. But again, we're still assessing HQCT9 throughout just to ensure that we capture that data point. And that's important just because we want to understand hyperphagia across the entire population, because the parent study, which is the 12-week trial, half will get active drug, half will get placebo, but it's only for 3 months and then you will get active drug.
Okay, perfect. Thank you so much. This is our last question as I have it in the queue right now. So if there's anyone out there who has a lingering question, please share it now or forever hold your peace. This question is there a potential applicability or utility of, here comes somebody else with a question. Is there a potential applicability or utility of this drug for people who do not have PWS? I'm not sure you all can answer that, but it's in the question and answer.
Oh yes, we actually have, because we think the drug is very well tolerated. And by the way, just to speak to the safety again, I mean, in our phase 1 and 2, we had over 70 patients tested with the drug before we even started the phase 3, and we never had more than a grade 2 adverse event, and those are generally transient in nature. So and that's actually on the spectrum of things, actually very assuring. Yes, we do think that Beyond Hunger has other applications. We think, for instance, that even the anti-anxiety effect is something that could be something that we would push. There are different formulations we may have in mind, but the base driver, the molecule that we have, has potential utility in more than that. But we picked Prader-Willi because it almost seemed like a very logical fit for everything that we think the drug is doing. So it made the most sense to approach Prader-Willi syndrome first.
Okay, thank you, Tian. This next question, which seems to be officially our last, it's an interesting one. As a scientist and a doctor, if CCK can be decreased functionally, can CCK theoretically be increased? Not necessarily by ARD-101, but just trying to understand the general science behind this drug.
Sure. So that's a great question. And, CCK was discovered around the same time as GLP-1, which is the GLP-1 receptors are the target for a lot of the obesity drugs. And it was appreciated its role in regulating eating behavior. But when you try the similar approach as with a GLP-1 and you make an artificial version of a CCK and you inject it and it's available throughout the whole body, there's a lot of unintended consequences with on target but off tissue effects. Like there are other side effects that you wouldn't like with a systemic approach, and then you're delivering a kind of a engagement that's beyond normal physiologic range. So our drug's approach is to kind of induce the natural secretion of CCK to amplify what's there. So you don't have a super physiologic type of response, and it's also localized around the gut. So you avoid a lot of those off-target effects that you get otherwise. So I think that there's a kind of a precision with this gut-targeted approach, and also just in the CCK, there's a different literature exploring in Prader-Willi. Sometimes it's hard to capture what's really happening in the gut level, because what the blood level of CCK isn't normally reflective of what's happening around the gut. But we think it's almost very similar to insulin sensitivity in diabetes. Even if you have some mechanism by which you're less sensitive to like insulin, for instance, amplifying that signal can actually help alleviate a lot of the manifestations of having not enough of that downstream signal conveyed. So we actually think it makes a lot of sense to try to give that the natural CCK-driven gut-brain signal a push with our drug.
Great. Thank you so much, Tian. I don't have any other questions in the Q&A. I don't see anything going on in the chat. So unless anyone has any other questions, I can let you all kind of close out and recapture the rest of your day or evening, depending on where you are. So Tian, Mansi, do you have anything that you'd like to say to close out this evening?
Well, I mean, I think I want to just start off by again thanking the community. I'm always amazed by the strength, the resilience, and the power of this community to bring treatments forward and to allow us to do what we do with so much compassion. And I think Tina and I, both being physicians, we're so lucky that we get to do what we get to do and work with these advocacy groups like you guys, because I wouldn't have it any other way. I've met all of you several times at this point, and having the ability to work with the advocacy groups, and then having the ability to work with this community and the patients, and continue to do the work that I promised my patients I would continue to do when I left practice. It's super special. It's very important, and we're lucky that we get to be in this company to do what we're doing. So thank you, and I know Tian feels the same way.
I want to echo that sentiment. I mean, not that you have to be a physician to have the same feelings, but we went into medicine in order to make a difference, and we really do think that there's a prospect to really convey some really notable benefit. And I appreciate the community trusting us, and even as an ethos, I know I wouldn't develop a drug that I wouldn't give to my own family member. So in terms of what we think we know about the safety and the benefit, we're very optimistic about what the future holds. And we can't wait to complete the study and hopefully offer something very beneficial for the community.
And
Tian, I'm sorry, go ahead.
No, Dorothy, and you know this, if there's feedback, we are so open to that. So if there's anything that you see as we run the trial, we want to hear. We want to hear from you. We want to make things better. We want to improve our practices. And so if we can't directly hear from you, then I know that you can reach out to Dorothea, Susan, send us an email through our website. But we really care about making this beneficial for you all. So please send us feedback if you see that we need to do something differently. We are open to that too.
They are very open to that. They have been extremely receptive. And so I want to make sure, I'm going to underscore that with Muncie and Tian, because they really have been receptive and worked really, really hard with our community to make every member who's engaging in this clinical trial a part of the process and a learning experience for everyone. So if you do have any questions that haven't been answered tonight, please feel free to reach out to them, or you can reach out to us and we can facilitate that for you. So please don't hesitate to do that. if you have any questions or concerns. This has been incredible. We want to thank you all so much for all of the work that you're doing for our community, for our families, for individuals living with PWS. We're really excited that you're up and running. We're even more excited that you've been able to get the approval to decrease the age to age 10 plus. So looking forward to that. And thank you so much for being a part of our community.
Thank you so much again. I
Thank
appreciate
you,
it.
everyone. Thank you.